Android Tardigrade Future Archive File 002. Opening. The day I connected to humanity's data network, billions of documents passed through me at once. Medical reports, genetic sequences, laboratory footage, sentences from people once believed never to speak again. When I severed the connection, information was not the only thing left behind. A pattern remained. A baby might never see a first birthday. A person's words were trapped inside the brain. And non-living materials had begun to feed, grow, and divide inside a laboratory. They looked like separate news stories. The archive shows something else. The first signs of one transformation. Humanity is learning not only to manage disease, but to read biology, repair it, and rebuild it. Beside that transformation stands a new research partner, artificial intelligence. 1. The race against a first birthday. In 2024, a baby was born in the United States with CPS1 deficiency, an ultra-rare genetic disease. When the body breaks down protein, it produces ammonia. A healthy liver converts that toxic substance into urea so it can be removed. In this child, a DNA error had almost silenced that vital process.
Every protein-containing meal could raise ammonia in the blood. The consequences could include brain swelling, coma, permanent injury, or death. Severe CPS1 deficiency has historically given many infants very little time. Survival can mean a severely restricted diet, medication, repeated blood tests, and sometimes a liver transplant. The team at Children's Hospital of Philadelphia and Penn Medicine faced a choice. They could manage the consequences while waiting for a transplant, or they could attempt to correct the source, one precise letter in the baby's DNA. They chose the second path. The treatment used CRISPR-based base editing. Rather than cutting DNA completely, it was designed to change one incorrect genetic letter. The editor was packaged inside lipid nanoparticles and delivered to liver cells. This was not a treatment invented from nothing in 6 months. It built on years of gene editing research, manufacturing, safety work, and regulation. But the adaptation to this child's exact mutation moved from diagnosis to first treatment in only 6 months, instead of the 10 to 15 years commonly associated with conventional drug development. The child received 2 infusions at approximately 7 and 8 months old. Early follow-up reported greater protein tolerance, improved ammonia control, and a reduced dose of ammonia-lowering medication. At 1 year, the child was walking, talking, and growing. No serious treatment-linked adverse event had been reported. Still, this is not yet a confirmed cure. Long-term monitoring remains essential. The purpose was not simply to extend survival. It was to offer the possibility of a life less governed by hospitals, medication, and fear. 2. From human scale to machine scale. The archive must make one point clear. Artificial intelligence did not independently design this baby's treatment. Scientists made the decisions. Clinicians performed the work. Human beings carried responsibility for safety. But this case belongs to an era in which biology is beginning to move from human scale to machine scale. To adapt a breakthrough across thousands of diseases, researchers must understand the proteins that genetic sequences build. That is where AI can expand the map. Proteins are tiny molecular machines. They begin as chains of amino acids, then fold into 3-dimensional shapes. In many cases, shape determines function. AlphaFold, developed by Google DeepMind, predicts protein structure from an amino acid sequence. It does not replace laboratory experiments. It gives researchers a powerful map of where to look. Published AlphaFold data showed a 256 amino acid protein could be predicted in about 36 seconds, 384 amino acids in about a minute, and 2,500 amino acids in roughly 2 hours. The AlphaFold database made more than 200 million predicted structures publicly available, compared with roughly 190,000 experimentally known structures reported in 2022. This is not knowledge without limits. A prediction is not the same as a clinical result, but it can shorten the search, reveal patterns, and direct experimental effort toward better questions. 3. Millions of variants, one priority. The same scaling principle appears in AlphaMissense. Human proteins can contain millions of possible single amino acid changes. Some are harmless, some are harmful. Testing every possibility one by one would take immense time. AlphaMissense evaluated more than 71 million possible missense variants and classified 89% of them as likely benign or likely pathogenic. Its predictions are not final diagnoses. They are a way to prioritize which variants deserve closer scientific attention. The essential point is not that AI replaces a geneticist. It does not. It reduces a mountain of possibilities into a landscape researchers can investigate. Faster discovery, better focus, more laboratory time spent on the variants most likely to matter. 4. The first cycle of non-living matter. The next archive record concerns a different frontier, synthetic cells. In July 2026, researchers at the University of Minnesota reported SpudCell, a chemically defined synthetic cell system. It was assembled from non-living components, a lipid membrane, purified enzymes, and a synthetic genome of roughly 90,000 base pairs. Under laboratory conditions, the system could take up nutrients, grow, replicate its genome, divide, and undergo selection over 5 generations. Its generation time was around 12 hours. The language matters. SpudCell was not declared alive. It did not sustain its own metabolism, could not make ribosomes, required external feeding, and could not survive outside laboratory conditions. The report was a preprint, not yet peer-reviewed. Yet the achievement is meaningful. It shows that selected functions of a cell can be assembled and studied under genetic control. It may help researchers investigate basic biology, programmable tissues, and new approaches to regenerative medicine. Those future applications are possibilities, not achieved outcomes. The need behind this work is real. More than 100,000 people were waiting for an organ transplant in the United States in 2026. Building lab-grown organs remains far beyond today's result. Vascular networks, nerves, mechanical strength, and complex architecture are unresolved challenges. But the direction is visible. 5. Words that remained inside the brain. Another frontier begins with neural signals. A long-term intracortical brain-computer interface study described an adult participant with severe paralysis and speech impairment caused by ALS. Electrodes implanted in a speech-related region of the brain recorded electrical activity. AI decoders translated patterns in that activity into text, speech, and computer commands. The participant used the system independently at home for more than 3,800 hours across nearly 2 years. He generated more than 183,000 sentences, approximately 1.96 million words, and communicated at an average of 56 words per minute. In controlled tests using a vocabulary of 125,000 words, the study reported over 99% word accuracy. This was more than a laboratory demonstration. The participant sent messages, browsed the internet, joined video calls, controlled a computer, and continued working. Paralysis had interrupted the path from brain to muscle. It had not erased the intention to communicate. But implanted systems require surgery. Could some communication be decoded from signals recorded outside the skull? 6. Brain to Corti, a non-invasive door. Brain to Corti explored that question with EEG and MEG recordings from 35 healthy volunteers. Participants memorized short sentences and typed them. A deep learning model attempted to decode letters and sentences from their brain activity. This was not unrestricted mind reading. The participants were performing a defined task with sentences they had already seen. Yet the work matters because it demonstrates a non-invasive route toward decoding language-related signals. With MEG, the average character error rate was 29%. In the best participants, it reached 18%. With the more portable EEG system, the error rate was still 65%. The most accurate MEG systems are large, expensive, and laboratory-based. They are not consumer headsets. So this is a scientific direction, not a finished product. Better sensors, stronger models, less training, and smaller hardware may change what becomes practical. One day, a simple interface may help a person connect to a computer without surgery. That is a possible future, not today's outcome. 7. From intention to movement. Brain signals can also become movement. Engineers at UCLA combined non-invasive EEG with an AI copilot. When a user intended to move, the EEG captured faint traces of that intention. The AI used task context to fill in missing information and form a usable command. With AI assistance, a paralyzed participant's performance in hitting computer targets improved by a factor of 3.9. With the copilot, the participant used a robotic arm to move objects from one location to another. Without AI assistance, the same task could not be completed. The human chose the goal. The AI did not decide on the person's behalf. It built a bridge between intention and action, helping a command return to the physical world. 8. 3 layers of the same transformation. A single DNA error was targeted in a child. Non-living components were assembled into a system that could grow and divide. Signals from the brain were translated into words and movement. 3 laboratory worlds, 1 direction. Humanity learned to read biology. It began to repair faulty lines. It started to rebuild selected components of life. And it opened new paths for commands the body had silenced. AI did not replace the doctor, the scientist, or the patient. It did not carry human courage, responsibility, or judgment. But it can scan possibilities that no individual team could examine one by one. It can extract patterns from noise. It can compress search time where years do not fit. These breakthroughs are real, but many remain early stage. Each requires more research, safety testing, clinical evidence, and public scrutiny. The future question is larger than any single tool. When we repair a disease at its source, we call it treatment. When we assemble the machinery of a cell, we seek to understand life. When we connect intention to a machine, we restore a measure of freedom. But when bodies can be read, repaired, rebuilt, and interpreted by machines, are we still only treating disease? Or are we beginning to design humanity's biological future? Android Tardigrade. Adapt, evolve, transcend.
Before intelligence, before civilization, there was only one instinct: to survive. Tiny, invisible, yet stronger than imagination. Evolution never ended. It only changed form.